All right, everyone, good afternoon. Thanks very much for sticking it out. There's still a lot of people here, very few empty chairs, which is great. I know that we've all got a super busy schedule for ARVO, and a lot of us have meetings this afternoon and evening, and the ARVO Gala starts at 6, and some people are going to be attending that. So I wanted to get us started here for this closing and summary session so that we can all get out of here by 5 o'clock or before. So thank you all very much for participating. I was only there for 20% of the conversations, but I thought it was fantastic in everyone that I attended, and hopefully you felt the same way. I have this dilemma that I don't know what happened in the other 80% of the conversations. So we wanted to take a little bit of time here, about 45 minutes, just to kind of provide some summaries of all of the discussions. And obviously we can't possibly go into all the detail. So this will be a very high level, hitting the highlights kind of presentation, led by the moderators, but we have 2 handheld microphones as well. So we want all of the participants to participate if you'd like. Basically, the idea here is we're just going to go through the subtopic discussion groups one by one and kind of tell each other, let us learn from each other, what were the highlights, what were the most important points to come out of this, and where do we go from here. So without further ado, I'm going to just start us off for subtopic discussion group number 1, which was on RGC differentiation and development, that were moderated by Carol and Tom Ray and Don. So Tom Ray or Don here, and either one of them want to provide some comment. Yeah, they both were not here. Okay, great. Well, so I wasn't there for any of those, but anyone that was there for any of the groups in subtopic discussion 1? Yeah, all right. So they're still here. They're just got it. All right, excellent. Well, maybe Brad, do you want to? Is Brad here yet? All right. Well, we'll come back to that one too. I know that Peter and Mara are here, so you guys. That's fine, yeah. Well, the only thing is we want the people on Zoom to hear. And so how do we turn our mics on? There's a switch on the bottom. I don't see, oh.
Yeah, thanks. So the topic of our subject discussion group was on RGC survival, maturation, and post interactions. The topic that we discussed was what models and tools are most useful for better understanding of the molecular mechanisms that contribute to death of endogenous and endocyclic ganglion cells. We had a great introduction from Dr. Singh and Dr. Hopkins regarding models and things to consider when we're looking at.
We can't hear on Zoom.
Does anyone from the room, I mean, what you've heard from me all day, does anyone from the room have any particular topic that they were down by, or that one question that they thought they might want, the expertise of the group that may not have been present? No, not hearing. They still can't hear.
How do I plug the audio into this? Do we know? All right, Mara, I think you're gonna have to just come up here. Can the people in the Zoom hear me right now when I'm standing at the computer?
Yes.
Okay. So we'll just have to have people come up here. And then if people talk on the mic, just repeat.
Okay, yeah, no problem. So I thought one of the more interesting, I personally have been a rodent model and then moved to a non-human primate model, but we had a lot of interesting, stimulating conversations regarding the pig model of glaucoma, a microbead model, and other smaller primates. And does anyone have anything they want to add to that, or do you just want me to? Trying to look for the gentleman from Denver that brought the pig model up, and I do not see him. Maybe he is in the room. Anyone? Gosh, trying to moderate online and in person is difficult. It's like
corralling my kids in the weekend.
So I'll ask you a question, because in our groups, we discuss primarily rodents, and then we talk about non-human primates and tree shrews. But one of the very interesting things that came up that Brian Samuel shared with us is that tree shrews apparently have excellent optics. For whatever reason, they get this sweet spot with size of the eye and refractive powers of all different structures, such that you can get really beautiful images of the cells in the retina, even without adaptive optics. We didn't talk very much about the pig, so I am interested in what was discussed about pig models.
See, it's
I'm not going to
like
repeat all of that.
juggling.
Right, so we talked primarily about rodents and non-human primates. But I'm interested because group number 2 didn't talk very much at all about pigs, and specifically, what disease models do we have in pigs, and how do they relate to the existing disease models for optic neuropathy in other large animals?
I could be wrong, but I believe the gentleman that had brought it up was talking about a bead model in pigs, and that because they would induce elevated intraocular pressure in the pigs, and that they were able to do more of the functional endpoints that we were looking for to see if we had successfully transplanted retinal ganglion cells, and that this might be a cheaper intermediate or maybe even precursor to human studies, because you have a functional analysis. So yes, we're interested in synaptic connections. Yes, we're interested in cell survival. But if you give the cell, if you transplant the cells and you get a great functional correlate, that might be some indication that it's working, and then go backwards and look at the molecular mechanisms of it. And on that note, not to move from your pig model question, because it's really outside of my area of expertise, but we spent a good amount of time discussing it, is the question became, when we discussed functional analysis of whether we had efficiently transplanted retinal ganglion cells in any model, the question was, how many do we actually need to transplant to produce the physiologic and functional endpoints that would be significant to correlate to human studies? And as far as our room knew, no one had ever done that dose-dependent model. And so I pose it to the group. And is there anyone have any particular interest in a healthy retina versus a diseased retina, and would that change?
Yeah, I actually want to ask Harry, who's having a conversation back, but Harry, you've done a lot of work in monkeys, looking at correlations between, and in humans, quite frankly, look at correlations between visual field loss, levels of vision, and the numbers of ganglion cells that survive at a given time. So based on that, do you have any back of the envelope ideas or calculations of how many ganglion cells we might need to make functional in order to support some rudimentary level of vision in humans?
The only quantitative thing that we've ever done looked at how many ganglion cells were remaining in a foveal area of glaucoma patients who still had 20/30 vision. So this is a patient whose medical history was known. We counted the ganglion cells in their retina post-mortem. And do they have 20/30 vision? And the number, with a calculation based on it by Lars Friesen, looking up the papers by FRISEN, Lars Friesen and me, was 5,000 ganglion cells to have central vision at that level.
Wow.
Now, whether that corresponds to how many ganglion cells somebody who's NLP would want to be able to walk through the corridor here and not walk into chairs, I don't know.
Yeah. That's interesting. Just to repeat for the people on the Zoom, post-mortem histological studies in humans have shown that in the fovea, people with 20/30 vision, although probably just a central island, may have as few as 5,000 ganglion cells to support that level of vision. That's very encouraging, I think, for our work. May not need as many functional ganglion cells as many people have thought.
Somebody just asked, how many does a normal human have? And the average is about 800,000.
Okay. How many RGCs does the normal human have? About 800,000. And it varies by about an order of magnitude, anywhere from 300,000 to 3 million.
The group was primarily talking about what number would we need
Right. Yeah, if you get 5,000 to connect, that sounds doable on the face of it. Great. Any other thoughts about this, the discussions for group number 3? Yes.
I'll bring it up and you can repeat it if it's short. I think something that came up in almost every session that we did, and then I think it's come up in a lot of sessions, is when do we do the transplantation? Because Leonard B made a great comment that to really see the psychophysics of human, we probably need a true baseline, which is 0 retinal ganglion cells, like to see a signal, but that might not be the best time to actually do a transplantation.
Yeah, I think that's a fantastic point, and it came up in our session multiple times as well. What types of models and at what disease point should we be studying? And when this comes time for human clinical trials, who are going to be the first people to get stem cell transplants or RGC repopulation? And for the people on the Zoom, the point that was made is from a safety perspective, from an ethical perspective, you might assume that patients with no light perception vision should be the first ones to get this. And from a perspective of being able to capture with good signal to noise, functional improvements in vision, you want people with very end stage disease. But those people are going to have the worst protoplasm, extracellular matrix, recipient environment in order to actually get functional reconnectivity. So there's probably got to be some kind of a compromise there.
Well, I may be jumping the gun here. This is sort of approached in our topic 5.
Yep.
We were looking at clinical transplantation, but potentially entertaining other optic neuropathies that are not glaucoma, like traumatic optic neuropathies, and some of them optic glioma, where they have this insult. It happens with line and then it's smolders and burns out. That potentially being the patient population that could be further risk of retinal ganglion cell transplantation. See a visual outcome that we would expect to see anything.
Yeah, I think that's a great point. That was another theme that came up multiple times. Aside from just thinking about the disease state that we should be studying, both in experimental models and in clinical translation, what diseases should we be studying? And I think most of us have the most experience and are thinking most frequently about glaucoma because it's so highly prevalent. But there are a number of changes to the lamina cribrosa and the optic nerve head and within the inner retina that may make it especially difficult to really reconnect the eye to the brain in that disease. So as Mara was mentioning, and I heard from other speakers in our session throughout the course of the day, it may be better to think early on about other diseases like traumatic optic neuropathy, optic glioma, or something where you get a relatively quick degeneration of RGCs, but with less chronicity and less other changes to the non-RGC subtypes in the retina. Does anyone else have any thoughts about that? And specifically, are there specific diseases or specific experimental systems that were are potentially not being thought about enough right now?
Wondering about retinal arterial.
Yeah.
Where you use, most cases of 7 are essentially blind. So that's something we're talking about.
Yeah, that's a good question. So Tom Ray asked about retinal arterial occlusions. We didn't get into that so much, and it's an interesting thing to think about. I think that may not be a pure RGC insult, and the other interretinal, internuclear layer neurons are probably affected by that. But arteritic ischemic optic neuropathy is one that Len Levin brought up, which happens very quickly, leads to profound retinal ganglion cell death and profound loss of vision, and might be a particularly good disease in which to study, assuming that there's not a complete ischemic insult that is permanent and therefore wouldn't be able to support metabolically the RGCs that we're trying to repopulate. Yeah, why don't you want to come up and talk a little bit? No, please, yeah. We didn't talk too much about age, but age is absolutely something to consider.
Yeah, I think that's pretty much it. Someone had brought up
Staying on glaucoma and that patient population still needing some assessment, that some therapeutic endpoint for restorative vision, and thinking about some of the more juvenile glaucomas that might give us a younger patient, but still allow us to transplant in patients with glaucoma, or those with uveitic, which to me sounded a little more complex than I was willing to entertain, considering the role of microenvironment and immune cells in restorative or successive retinal ganglion cell transplantation.
Yeah. The other thing that came up with regard to age is the experimental models. And this is true of both age, but also the degree of optic neuropathy that we're looking at and looking to study. I mean, Brad, I think you made the point that if you want to take a macaque and elevate the IOP and wait until they have advanced end-stage disease or no light perception vision, that will take years. And that's incredibly expensive to wait that long, both from time and dollars. So that makes this tricky, but I think it's important because we want to be experimentally studying systems that are analogous to the patients that this will be translated to. All right, that's great. I do see that Don and Tom Ray are back. So I don't know if you want to come up. I'm encouraging you to come up, but if you don't want to, unfortunately, because of the Zoom people, we do need one person at the podium. Great, Don's on his way. Yeah.
This hook up directly?
Let's see. Oh, you want to plug in? You made slides. That's very impressive. Yeah, you really set the bar here.
I'll hold it up.
Are you on the Zoom now? That's the only issue.
I don't know. I haven't seen it yet. Actually, it's tricky. Can we show this without it being on Zoom? Because then I'd have to find that.
All right, we're gonna.
All
I'll
right,
tell you what I'm gonna
we'll
do.
do it after picture.
No, that's fine. You full screen it. This is gonna be. Sorry, this is clearly the least organized part of this meeting. But
How do I get that?
I'm gonna just put my camera on it and hope that the people on.
How do I get this to show?
Oh, full screen it. It will, but you just gotta go to full screen. Let's see. Sorry for everyone on the Zoom. That's the best we can do.
So our group was RGC development and differentiation, and especially when I think the scribes, and we look forward to how they're going to put together our sort of interesting but random circular kinds of discussions. But this Venn diagram here was trying to separate two different aspects of RGC subtypes. So we spent a lot of time talking about what are the human retinal ganglion cell subtypes and how are they different than mouse. And we talked about the issue of, from a lot of groups, single cell work. It looks like the mouse probably has more different ganglion cell subtypes than the human and non-human primates. And yet at least the way we think of it, our visual system is much more sophisticated and complicated than the mouse. So why do we have fewer subtypes to do more complicated practices? So from the point of view of developmental neurobiology, we want to know what are the human RGC subtypes? How can we generate them in culture? Because we are not very good so far generating subtypes. And even cell maturity, how do we get ganglion cells in culture to be more mature? How do we define mature? And again, how do we generate the subtypes? But that we think is actually a little bit different than optic regeneration clinically translated to actually helping patients see what subtypes do we want to make. And some of the other groups have talked about that. I think that was redundant. But we started thinking about it. It's not even a subtype. We want to define what is the ideal type of ganglion cell for transplantation. And that doesn't have to actually be an existing subtype. So what are the characteristics if you were made for to cell? We want to talk about survival, and several groups what does it take to get this beautiful cell on the right to survive? Axon outgrowth, which is really key, and then even harder is guidance in synaptogenesis. So if we could come up with a wish list, what is the perfect ganglion cell for transplantation, even if it doesn't exist in nature, can we generate that? And then something we talked a little bit, but not that much about, is quality control. You know, if we're ever going to actually put these cells in patients to get cells that are really reproducible, because even in the same lab, each person who makes cells is different. Each prep is different. So what are the quality control measures that are needed? And then get back to this translation. Again, we talk about better defining the subtypes. What is the ideal cell? How do we make them resilient? Because we know from a number of studies, people in this room and elsewhere, that different subtypes are susceptible to different kind of injuries. And glaucoma versus optic glioma versus mitochondrial optic neuropathies, it's not always the same. So different cell types have different survival characteristics. So if we can understand better how ganglion cells die and why some types are resistant, maybe we can engineer that perfect cell that's going to survive and do great things. And then we also talked about various CRISPR, other kinds of screens that other people are doing, as well as our lab, to try to improve. Can we improve by nature? And the last session, which I guess didn't get on our slide because we're already late, is really looking at transdifferentiation. And Tom's lab and others have really done a good job of, what about endogenous regeneration? How we take the newer cells and regenerate them into ganglion cells. Do other members of the group or anybody else want to add? Related to survival, we felt that survival was necessary. It's obviously getting cells have to be there, but it's not sufficient. We talked about some examples like knockdown of some survival injury factors like DLK, LZK. We can get cells to survive, but it makes them less good at regeneration. So it's a balance of survival and regeneration, but not the same. And sometimes they're actually going opposite directions. So that's going to be tricky. Any comments from the group or anybody else?
Thanks. All right, back in business here. Well, I guess now we see why Tom and Don were a little late coming into the meeting, but if you're going to be overachievers, then I think you're perfectly excused. That was really nice. Thank you very much. All right, so now SDG 2, Brad has very kindly agreed to do a very quick off the cuff description of the things that we talked about.
Apologize for being late. I was making slides, but one of my kids spilled his chocolate milk on them, and then my dog ate them. So I don't have slides. No, I was having a really good conversation with one of the attendees from the fifth subgroup meeting about ILM. So I'll work our way backward. We, in that final session, were talking about getting past the island, getting cells past the island in particular, and all of the different approaches that have been tried or that might be tried, from enzymatic digestion. Tom has done a lot of work in this area. We heard about staining the ILM with ICG, which has already been used surgically in human beings, FDA approved approach, and then using a laser to create vaporization bubbles that mechanically poke holes or uniformly thin the island. All very interesting approaches. We talked about surgical approaches and had the expertise of a vitreoretinal surgeon from Gießen, Germany, informing us a great deal about how difficult that will be to make an intraretinal or a sub-ILM injection. Although that's been published, there's a great deal of concern that this will be a viable approach that in essence, you'll wind up basically being subretinal with delivery in that regard. So it was a great session, and I think highly practical in many ways. This is going to be really important when we consider different models. I guess I'm jumping now all the way back to our first session. We talked about different species and different approaches to creating experimental glaucoma across different species. And in that context, you have to think about the really, really small eye having some advantages. For example, the high numerical aperture that makes for astounding imaging, being able to see neurites outgrowing on donor cells implanted into a retina. But on the other hand, you can't do anywhere near the kinds of surgical approaches that might be ultimately required, such as placing cells in the subretinal space efficiently, effectively. We heard from people at the NIH telling us how you really, if you're going to create a custom tool for delivery and you expect to translate this at any time, you'll have to go through regulatory to have so many aspects of that tool approved and characterized and documented, and that ideally you should also have IP attached to that, because to get through all of that regulatory, you're going to need a lot of industry money, and without the IP attached to it, you'll never get the industry money. So it was a wide-ranging conversation. One of our sessions had to do with, you could say detecting, but more like characterizing the donor cells. Can we visualize them? Can we measure their function in various ways? I think we sort of landed on the idea of using a combination of fluorescence detection techniques, such as fluorescence SLO, or even simple fundus cameras that are outfitted with the right emission and barrier filters in order to detect fluorescence, but then in combination using OCT with its axial resolution, and that combination in order to both study the integration, anatomical integration of the transplanted cells, but also their whatever effects the transplantation has had on the host retina, from toxicity to mechanical effects, et cetera. We talked about optophysiology and the ability to see functional light responses or optogenetics and being able to detect functional light responses at the single cell level in the donor cells. We were talking about how Ben Sevier's lab will be doing some multi-electrode array recordings ex vivo, and Jonathan Susi brought up this fascinating new technology whereby you can have an in vivo sort of thin fiber sheet that's been apparently used now somewhat more extensively in brain, but apparently also in retina, whereby you can do essentially multi-electrode array recordings in vivo. So that was astounding. And in that regard, we probably need to use DREADS and CNO or again, some way of identifying the donor cells, if not by fluorescence. If you're doing a strictly functional recording, you need to be able to elicit the specific responses from the donor cells. So, to be able to detect their function and thereby their functional integration in vivo, ideally. Because again, you won't be able to do this when we get to translation. That's the point. You won't be able to do that in the human being, to just take the retina afterward and be happy that there was some functional integration that you detected by histology and by electrophysiology. You'll need in vivo techniques ultimately when we get to clinical trials. And so we talked a great deal about imaging, adaptive optics, two photon microscopy approaches. And what was the other session actually was also overlapping with, I'm sure, a lot of the other groups, and that was host and donor cell interactions with regard to immune functions, adaptive immunity, innate immunity. Peter Baranoff told us that most of the data are pointing to the majority of cells, donor cells implanted, really succumbing to innate immune cell reactivity. and how we talked a great deal about how we might sort of find the right balance between suppressing that without losing the beneficial effects of microglia, for example, in the retina, and minimizing interruption of normal retinal processes in that regard. So we have to find this balance. There was also some degree of discussion about systemic immunosuppression. What are the right protocols? What, without getting dangerous toxicity from overdoing things. I'd say the theme of the day was that we raised many more questions than we answered. And that, I think, is a great benefit of this group and this day. Thank you, Tom. Thank you, all the organizing committee members, because it identifies experiments that we all need to do. There was a common theme also that we all need to share our negative results so that we learn a great deal of what didn't work, and to not spend time and energy and expense on repeating something that someone else has already learned didn't work. I think, recognize that it's difficult to publish negative results, so that this kind of venue is really important for this kind of open sharing of things that otherwise don't necessarily get learned through the literature. What else? Is that 5? That's 5, okay. Questions or any other input from people who attended the session?
Question about surgical approach that you guys have. You mentioned both and and then I know that will be place, but there's some evidence that cells can be migrated to them. So
Yes.
the question is, did your group talk about relative, clinical applications for one
To some extent, and just to add to that, I had the pleasure to see some of these data presented recently in a Zoom through the Audacious Goals Initiative meetings, the consortium meetings, but also was discussed today. Jonathan Susie's using chemokines so that you can place the donor cells under the retina, but then in the vitreous have a signal to draw them to where they really need to be, which is another fascinating approach. I mean, the truth is we talked only broadly in the context of different models, different species, the smaller eye having its clear benefits from replicability, reliability, reproducibility of the model, high throughput, lower expense, including faster duration of study, shorter duration of study compared to some of the larger species and non-human primates, et cetera, and the greater complexity of those models. But in the context of surgery, just how much more difficult it would be on the super small eye to have those surgical approaches be successful.
Great. All right, thanks so much, Brad. That was great. Moving on, we've already done 3. So now SDG number 4, intraretinal wiring, which was co-led by Adriana DiPolo, Alex Klotkin, and Ivan. You have slides as well?
No.
Oh, okay. That's
But
good. That makes it easier.
Yeah. I'm very impressed by the slides. I'm very impressed by Brad being able to say everything without notes. I'm thinking, what? So going to be something in between. First, I want to thank Alex and Yvonne. They were fantastic co-moderators. It was a lot of fun to organize this session with them. And I also want to thank our scribes, Julie Cho and Michael Gilhooly. They did a fantastic job. They were like super intensely writing. And everybody who made introductions, Greg Schwartz, Liv Moons, Barb Burgoyes, I'm forgetting, Melanie Samuels, Tom, and Matt Van Hook for fantastic introductions. So our session was very challenging because we all realized how little we know about what happens in the inner retina during degeneration, even during development, although we know a lot more during development, but certainly we don't know enough during degeneration, let alone what we need to do to help these transplanted cells thrive in the IPL, right? So it was very helpful that Greg introduced goalposts. So what are the minimum and the maximum requirements that we should use to set what we are aiming for? So the lower limit would be just to put an RGC in the IPL and have it extend even a minute dendrite in the IPL. That would be like the minimum we could expect. And the higher limit would be to achieve synaptic connectivity, which is indistinguishable from the endogenous retinal ganglion cells, even for every retinal ganglion cell subtype with full functional recovery, right? That's like the idea, which we probably will never achieve. So we thought, well, maybe we can settle for something in between, you know, something that would allow us some level of success, which we to be able to program these retinal ganglion cells going to what Dawn and Tom were presenting, program some specificity to make connectivity in the inner and outer IPL. And so if we can generate generic on and off cells, and possibly with certain specificity for some bipolar cells. That would be fantastic if we could achieve that. And maybe we can combine it with visual experience that Alex was saying could help refine along the way the connectivity. But the truth is that very little is known about how can we achieve that. So we talked about intrinsic factors, what are the intrinsic programs that are required for this to happen. And we thought it would be really interesting to molecularly engineer the transplanted retinal ganglion cells to enhance their ability to integrate and rewire. And for that, it would be very interesting to have a single cell RNA-seq profiling or some molecular profiling of retinal ganglion cells that would allow us to identify, well, during development and during neurodegeneration, so that we could generate a detailed atlas that would guide us to understand what is required for these cells. But that would have to be done in non-human primates or humans to be valuable. So a common theme throughout the whole session was how mostly everything we know comes from mice and how little we know from non-human primates and humans and how important that is. So we need also powerful imaging tools to be able to track not only molecular changes, but functional integration and other structural aspects, arbors, synapses, and have a baseline during neurodegeneration to see what we have to overcome when we do the transplantation. One interesting idea that came out of the discussion was that sometimes we think a lot about development and how development recapitulates some of the things that happen during neurodegeneration. Many of us feel that we have to recapitulate development to repair the retina, the eye, the nervous system in general. And Leaf brought up the fact that retinal ganglion cells first extend the axons all the way to the brain. They make synaptic connections there, and then they develop these elaborate dendritic fibers. So in our case, you know, you have a cell that you put there and you want it to integrate and reconnect probably before it can have, it has the time to extend an axon and reconnect with the brain. So there was an experiment that came up, I can't remember exactly. It was sort of a group discussion where we thought about an experiment where we could take retinal organoids and I think Jason is doing a little bit of these with take a retinal organoid and you expose it to a brain target to induce retinal ganglion cell sprouting and then capture those changes by transcriptomics or some type of omics type of experiments. And then you can identify potentially some key master regulators or a set of genes that are required to induce dendritogenesis in the absence of brain connectivity. I don't know if that, I mean, that would be an amazing thing if it works, but an experiment to be done. We talked a lot about remodeling during neurodegeneration and plasticity and what happens when other cells disappear, when retinal ganglion cells disappear and other cells take over. Can we harness some of these plastic events to help rewire the transplanted cells? Can we understand the physiological plasticity in the retina during neurodegeneration, and to enhance and facilitate integration. So again, something we don't really understand about plastic changes and remodeling in the human retina during neurodegeneration. So I only have like 2 more points, so bear with me. We talked about extrinsic factors that I could go on and on. We'll talk about the ILM, and Tom helped define some of the limitations and opportunities about manipulating ILM. We talked about IPL sublaminar development and how amacrine cells contribute to these, mainly the sublaminar specificity come from amacrine cell signals and not retinal cells. So can we re-express or overexpress molecules in the amacrine cells to enhance these the connection? But again, we need a better understanding of molecular signals in the retina. We talked about the extracellular matrix components and how we need a systematic approach to map all the components and interactions that take place so that we can manipulate them and use them. We also talked about resilience versus vulnerability, like Don was talking about. And we wondered if we need to recapitulate the over 45 types of retinal ganglion cells that we see in the mice. And the consensus was that perhaps not, although that was a bit of a discussion. Do we really need to be so sophisticated? Can we just, as Don was saying, just generate the ideal retinal ganglion cell, which is resilient and survives perfectly well in the IPL? The problem is that if we focus on them being more resilient, does that also alter their ability to rewire and be more plastic? So there could be like a compromise between making a cell very, very resilient, and then it may not be able to respond or adapt well to changes in the environment. We don't know that. We talked about modulating the diseased in a retinal environment and how there's still so much that we don't understand about pathological processes, the longitudinal sequence of events that take place during disease, including cellular and molecular changes, and how that's going to ultimately determine the environment where these transplanted cells are going to be. That's going to be unique and change with the course of the disease and many things that we really don't understand, and how, again, human glaucoma tissue would be so helpful to understand and study. So we talked about how to modify the IPL environment, and we, of course, talked about growth factors, how these are probably the only type of molecules, neurotrophic factors, that promote survival, but we also realize that we don't want to, these are very potent factors that have pleiotropic effects that might be very tricky to calibrate and titrate in terms of the physiological levels that we need for them to be effective without causing some secondary or off-target effects. So it's going to be very difficult to tightly regulate the expression of growth factors in terms of levels, timing, duration, and target specificity. So those are things that we have to talk about. Okay, I'm almost done. And then lastly, we talked about the relationship between synaptic connectivity and successful function. So how do we measure success when we can finally make these transplanted cells connect and hopefully make synapses? How do we know they're working, and how do we measure success? Again, so a better understanding of normal connectivity, and Melanie Samuel was fantastic at providing some guidance in terms of understanding normal connectivity of retinal ganglion cells and how to measure success using transsynaptic tracing, nanoscopic imaging methods, and compatible with human tissue, and perhaps some functional outcomes that are available. And ultimately, how do we measure success in humans? So how do we know eventually when we are able to transplant these cells in humans, have integration? And what I guess we also laughed about the fact that when you just ask the person, are you seeing better? Can you see? So that's the beauty of doing this work in humans. Alex, do you have anything? I'm done, but maybe Alex wants to add something, or anybody who attended the session? Anything else I missed? I'm sure I missed a lot of things, but. All right.
Thank you so much. I think all of those points were very important. And what I really liked was the idea of this crowdsourced plans for new experiments, things they can answer, start to begin to answer some of these questions that are being posed. All right, the final subtopic discussion group is going to be number 5, and Kim Gakofsky is going to come up and also had help from Bill Guido.
Yes, I definitely drew the short stick here. I am Kim Gokowski, and I want to thank Bill, who brought legitimacy to our session, as well as our scribes, Mara Salem and Zhu Wei Wei, who helped scribe through our sessions. I'm gonna put the laptop in. So for our first question, we attempted to address the question of how can we get axons to regenerate long distances? And if you think you guys didn't have answers, we definitely didn't, because we were very far away from that. We thought that part of answering this question is addressing what the barriers are to long distance regeneration. And so they include addressing cell intrinsic and cell extrinsic barriers. In terms of the cell extrinsic barriers, we're very interested in understanding if anybody knew whether the guidance cues that were present during development that guided development of the optic nerve, chiasm, and tract are still there and the same in the adult, and whether they changed in the injured or diseased state. In addition, and similar to the developing tissue, in addition, we thought that perhaps we wondered whether the growth cones of regenerating axons look the same as developmental axons, and whether or not genetic modification gene therapy that we're using, the approaches that we're using, change the growth cones of these regenerating axons, and that if we're thinking about modulating the external environment, do we need to know what the growth cone of regenerating axons look like so that we can make sure that there isn't a mismatch between what we're instructing these cells to grow and do and where we're telling them to grow. Additionally, it's retinal ganglion cell survival, retinal ganglion cell axon growth, and retinal ganglion cell guidance appear to be separately regulated processes. And we're going to, of course, have to use combinatorial approaches to direct optic nerve regeneration. And the question arose as to, well, currently our approaches are to do all of this simultaneously, but rather would we achieve more success if we took a sequential approach with some of our gene therapy approaches. And then finally, we asked the question of what role activity plays in directing long distance axon growth. You know, we discussed Dr. Huberman's results a lot and the effect of forcing activity into the injured eye and whether that would play an important role in directing future therapies for optic nerve regeneration. For our second question, we asked what is the role that glia play in regeneration? And so of course, there are a number of different types of glial tissue in the optic nerve, the central nervous system, some unique to the optic nerve from the central nervous system. And as you all know, glia serve very multifaceted role in the optic nerve, including providing support for conduction, metabolism, waste cleanup, and sometimes they're helpful, sometimes they're hurtful, especially in the disease state. But despite this, we need to focus on modulating their function and how they can help, not just trying to eliminate or get rid of them. In addition, glia function and identity is constantly in flux. The interesting thing that we learned is that their identity is really dependent on the retinal ganglion cell. And so the neuron seems to be the driver of glial function. And so modulating or controlling how the neuron interacts with glia may be an important approach towards facilitating optic nerve regeneration. Major problem, though, is that glia are very difficult to transduce virally, and so they're just much more difficult to manipulate. My favorite line of the day from Nick Marsh Armstrong is, "You need to clear the crap before you can grow it back." And so I think that considering the role of glia is important, and maybe we should consider priming glial cells for injury. How could that be helpful in helping to clear some of the debris that might serve as roadblocks for regeneration? And then, and interestingly, in group studying regeneration in amphibians, glial responses appear to be transient, but the retinal ganglion cells readily regenerate. For our third question, how do axons navigate to appropriate targets? This question, of course, has been very difficult to study because we haven't been able to reliably regenerate axons to their targets. And so as a result, we really felt that the development of distal injury models was really important. And so especially work published by Dr. Hayes' group where the optic tract was severed just distal, just proximal to the superior colliculus, that development and standardization of these methods would be really helpful for actually being able to initiate more intensive studies into axon targeting. And then kind of similar to your question of retinal ganglion cell diversity, you know, retinal ganglion cell, and if we have to recapitulate that in the retina, retinal ganglion cells, of course, innervate multiple targets besides that within the primary visual pathway. And so we asked whether or not needing to target all of these other cortical diencephalon substructures was really necessary. And perhaps in our patients who are no light perception, just being able to regenerate to something like the suprachiasmatic nucleus to restore circadian rhythm would be a big win. And so we don't necessarily need to regenerate to every single target. Another major topic of discussion was how do I identify the functional synapses at a histologic level? Many of you guys, I'm sure, have done staining with VGLUT2 and PSD95. It looks like just sprinkled dots on your on your computer screen. And so kind of investing into using viral vectors to help kind of demonstrate synaptic connectivity between regenerated axons and targets in the diencephalon may be necessary. Behavioral tests in our animal models are really helpful because different behavioral tests such as the looming test or the visual cliff test, pupillary light response, and the optokinetic nystagmus all kind of elucidate different functions in different areas in the brain. And so being able to recover one function versus not another would be able to help us understand what's actually being successfully regenerated versus what is not. But also perhaps maybe setting those bars as signs of successful regeneration are too high, especially at this stage of our research. Maybe we need to focus on something simpler like the pupillary response or the novel object recognition test where an object is just placed into a cage and to see if the animal, most likely a rodent, can detect this new object and reacts. And then finally, we talked about subtypes of retinal ganglion cells and which subtypes regenerate. We know that there's literature to suggest that the A2 or the alpha-2 retinal ganglion cell subtype is more amenable to our regenerative therapies. But actually, it seems that combinatorial gene therapy approaches lend to a larger diversity of retinal ganglion cell regeneration than previously thought. Oh, I'm only on question 4. I thought it was done. So for our fourth question, what is the best model system for studying regeneration? I think we thought that this question really depended on what you were asking. So are you trying to recapitulate disease? Are you trying to recapitulate human anatomy? Or are you trying to find an organism that naturally regenerates and then to learn lessons from that organism and apply it to a higher vertebrate? And so with in vitro assays, you know, we don't really have a retinal ganglion cell line, right? And we all know that retinal ganglion cells readily expire in culture. And so limited by retinal ganglion cell death, it's really difficult to study axon growth. But I think we believe that co-culture experiments where retina is co-cultured with a cortical structure is underappreciated and should be revisited as a potential tool for studying axon guidance, as well as stripe assays. From an in vivo perspective, we admitted that we all use the optic nerve crush. We all admitted that it's not a great model system, but the advantage is that in general, it's you get a complete crush, and so it's generally consistent. An incomplete crush, you can kind of tell because we're generating axons that look different than spared axons. Partial crush is very difficult to reproduce, but has its own values. Transection is very hard, and then whole eye transplant is being investigated, but there are currently still difficulties with getting axons to regenerate past the transplanted globe. At a clinical level, we felt that glaucoma is still of the different types of optic neuropathies, the best model system to study because the injury is slow and not as bad or as intense as in other disease states. Finally, question 5, is there a critical period for regeneration? And so if we're talking about retinal ganglion cells themselves, of course there has to be, but we don't really know exactly what it is. If you think about optic nerve crush injury, you will see retinal ganglion cell soma death by 3 weeks. And so without other treatment, perhaps 3-week window is kind of where we are. In whole eye transplant group, they told us that with manganese enhanced imaging, they can still see axonal transport up into the transection site 3 weeks after transplant, suggesting that there is still some activity in healthy eyes that have been transected. But in part, this question of if there's a critical period really was, will there be downstream targets for these regenerating axons to synapse with? So in part, these retinal ganglion cells die because they are missing a synaptic partner, but that seems to be the case for downstream targets as well. Clinically, there have been reports of patients with advanced glaucoma that have demonstrated LGN volume loss, suggesting that there's anterograde transsynaptic degeneration. Other groups have studied this as well in the DB2JA model of glaucoma and seen LGN and superior colliculus changes 9 to 12 months after of life. And then finally, we believe that there should be more effort into studying amblyopia as a model system for understanding cortical plasticity, because once we all are able to regenerate the optic nerve, we want to know if there's really a bed for those nerves to connect with.
Wow, that was a lot of stuff. Great job. Thank you to all the moderators for summarizing that. Does anyone else have any final thoughts, reflections, anything that was particularly interesting or important that was mentioned today? All right, very good. Well, I'll wrap up here with just a few parting comments. I want to one more time just thank the supporters of this meeting, the consortium in general, and the EVS travel grants for having the forward thinking and generosity to support this mission. I want to thank everyone that was involved in organizing this meeting, the entire organizing committee, but also Brad, Alex, and Yvonne, who have come in and really put in a tremendous amount of work in the last 3 to 4 months to curate these questions that we all discussed today, and for leading these. And I also want to thank everyone that provided introductory comments for each of the discussion sections, and for the scribes for your hard work and taking notes all day. So as I mentioned at the beginning of the day, we want the RReSTORe Consortium to live on long past this meeting, for this to be a sustained endeavor. So hopefully what we've accomplished here today is just the beginning. We're going to be transitioning now to phase 3 of RReSTORe, which hopefully will last years. We're going to use the Basecamp website as our main platform for sharing files, thoughts, posting on message boards, asking questions, giving experimental ideas, asking if people have reagents that might be good for a specific experiment, anything that would be good to share among the group. It would be great to, we can do that very easily through Basecamp. So hopefully most people or everyone listening now has already gotten their account and password for this. If you haven't yet, please email Andrea Everett and she will be able to get you hooked up into that. And then I want everyone here to provide feedback. Please let us know what you want the consortium to be, because this is, you know, a group for all of us to maximize our collective ability to move the field forward. So things that we're thinking about doing in the coming months and years are ongoing collaborative discussions. I think it would be great to have virtual workshops and seminars. People can give talks on their research progress, especially if any of it is collaborative and has come out of meeting and interacting with other people at this forum. We might think about having SIGs and symposia from this group at future meetings of ICER, ARVO, SFN, anything else. But any other ideas that you guys might have, please let us know. One thing that I have heard, and I think it's absolutely true, is that collaborative science will be accelerated by seed funding. And I think that's something we're absolutely going to look into, is whether we can get small pots of money that would be able to spur collaborations between RReSTORe investigators to start some of the interesting experiments that have been described today. We're also going to be trying to assess the impact of this consortium on an ongoing basis. So we're going to give some time for the discussions today to percolate and for everyone to establish collaborations. But at some point in the future, I'd appreciate it if you could fill out a survey that will come to you that describes any new ideas, experiments, projects, or collaborations that come out of this. And especially for our EVSs in the audience, we'd really like to know whether and how much participation in this consortium has benefited you. And again, in the future, I think a lot of people have been discussing opportunities for publishing white papers or opinion articles, consensus statements on some of the topics. So that's something that we're absolutely going to think about doing. So look for emails in the future calling for volunteers for writing committees. The first step will be collating all the notes from the fantastic jobs the scribes did today. And then we'll post a summary of that on the Basecamp website so that everyone will have that information going forward. And with that, if there's any questions or comments, feel free to ask now. My email address is on the slide here. So please reach out to me if you have any ideas for the consortium going forward. And that's it. Thank you guys very much for your participation.